So this practical detection of sugar in urine is related to two chapters. One is biomolecules and the other is your excretory system. So excretory system, in that chapter you learn about the various parts of excretory system, like kidneys, ureter, urinary bladder, urethra. And in this chapter you learn about the how the blood is purified by the kidneys and the nephrons present in the kidneys and prepares the urine. Okay, so urine is the waste product which is formed after the filtration of the blood. It contains various waste things. But when any patient is suffering from a condition known as diabetes, then that patient is releasing the sugar in the urine. So objective is our objective is to detect the presence of detect the presence of sugar in the given sample of the urine. Theory is large volume of volatile and non-volatile waste products is produced by different metabolic processes in the body. So in our body, always there are many metabolic or chemical reactions are going on. So as outcome of those processes, the toxic nitrogenous waste is created in the body. So urine is a liquid byproduct produced in animal and human bodies. So it is produced in the kidney through a process called we can say urine formation, that process. And urination is the process through which it is excreted. So urination is the excretion of the urine through urethra, and it is produced in the kidney through the process of urine formation. It's not written over here. I think it is missing. So urine formation take place by the nephrons, and then the urination is the process of releasing, or micturition is the process of releasing of the urine. Now urination is the primary method of excreting. Through urination, when we excrete out the urine, we release the waste water, excess water, and with that we are releasing the uh, soluble chemicals, waste chemicals from the body. Let's discuss some characteristics of urine. Normal urine usually has light yellow color due to the presence of yellow pigment urochrome. So there is a pigment called as urochrome which gives urine its yellow color. Average production of urine in adult human is 2 liters per day. Depending upon various conditions, average release of urine is 2 liters per day, but it is subjected to the conditions. In conditions like temperature, like the food we are eating, like the amount of water we are drinking. So it may vary among in response to different conditions. The pH of urine varies between 4.6 to 8. So its pH can be 4.6 to 8, and the specific gravity of the urine varies between 1.010 to 1.40. Now let's see what are the constituents, what is there inside the urine. So now it's written, actually normal urine is highly complex aqueous solution of organic and inorganic substances. Urine consists of about 95 to 96% of water. The most important nitrogenous organic substance present in the urine are urea, uric acid, creatinine. So these are the nitrogenous toxic substances. Urea is also nitrogenous, uric acid is nitrogenous, creatinine is nitrogenous substance. So nitrogenous substances are toxic for the cells. If they increase around the cells can die. So they are it is necessary that they should be removed. The other organic substances are oxalic acid and lactic acid. So these are also organic substances, oxalic and lactic. You know, these are carbon containing acids, oxalic and lactic. And lactic acid is produced in the muscles during the heavy exercises. Oxalic acid is also produced because of certain food that we are eating. that oxalic acid is a metabolic waste which is produced in the cells. The principal inorganic constituent of urine are sodium chloride, potassium chloride, sulfates, and phosphates. So these are inorganic substances. Abnormal constituents of urine means abnormal when a patient is there, or when a medical condition is there for any human, then we say that the urine constituents become abnormal. And in the urine, along with the normal constituent, urine is also having sugar, glucose in it, ketone bodies in it, blood, protein, bile in it. So these are, we can say, abnormal things which should not be present, but because of the medical condition or disease, some type of disease is there inside the body. So therefore, the urine is having the glucose in it. Like diabetes condition. Diabetes, when a person is suffering from diabetes, glucose is not getting absorbed in the muscles in the liver, then it is released in the urine. Ordinary glucose sugar is absent in normal urine, but when the glucose level in blood exceeds the renal threshold of glucose, 160 to 180 milligram per deciliter. So when the glucose levels are very, we can say, high in the blood, the glucose is coming above the level which it should come. So glucose start to appear in urine. Then kidneys don't have any option other than to release it in urine. The presence of glucose in urine is called glucosouria, or glycosouria it is also known as. Both it can be called glucosouria or glycosouria. That means glucose is present, indicating the diabetes mellitus. And we can do two tests to test to be confirming that the sugar is present in the urine. We can do Benedict test or Felling's test. So these are the tests which can be done for presence of sugars, detection of sugars also. If you want to detect that any sample, given sample is sugar, for example, I have given you some crystals and I want to tell you which is that substance, you can do Benedict test and Felling test. Sugar in urine. Benedict's test. Materials required: test tube, test tube holder, urine sample, measuring cylinders, Benedict's solution, and burner. Procedure: take 2 ml urine sample in a measuring cylinder from the urine sample bottle. Take a test tube and pour the urine sample in it. Take 5 ml Benedict's reagent in a measuring cylinder. Add Benedict's reagent to the test tube that contains urine sample.
Using a test tube holder, hold the test tube firmly and heat it for 2 minutes on the burner. Keep shaking the test tube while heating. A yellow precipitate appears, which indicates the presence of sugar in urine. Depending upon the concentration of sugar in the urine, either green, yellow, or brick red precipitates are formed. Fellings test. Materials required: test tube holder, urine sample, measuring cylinders, filling solution A, filling solution B, and burner. Procedure: take 2 ml urine sample in a measuring cylinder from the urine sample bottle. Take a test tube and pour the urine sample in it. Take 2 ml filling solution A in a measuring cylinder. Add filling solution A to the test tube that contains the urine sample. Take 2 ml filling solution B in a measuring cylinder. Add filling solution B to the test tube that contains the urine sample. Using a test tube holder, hold the test tube firmly and heat it gently for 2 minutes on the burner.
So in the urine sample, we have in this Falling test, we have taken 2 ml of urine sample and then 2 ml Falling A and 2 ml Falling B, and then we are going to put it on the to heat it on the flame and to
Keep
observe
shaking
the
the test tube while heating.
change
A
of
green
color.
precipitate appears, which indicates the presence of traces of sugar in urine. Depending upon the concentration of sugar in the urine, either green, yellow, or brick red precipitates are formed.
So, on the basis of which color precipitate is formed, green, then it means the percentage of sugar is less, and when the sugar is increased in means if it is more in the sample, it will be yellow, and if it is very much 2% or more sugar, then it is red color. Okay. Now simulator, now video is there. You watch this also. And the lab, actual in the lab, how it is done.
Detection of sugar in urine. Sugar is ordinarily not present in normal urine. When the sugar level in blood rises above normal level, the kidney eliminates the extra sugar through generally Benedict's and Fellings tests are used to detect the presence of sugar in urine. These tests give colored precipitates such as green, yellow, and brick red, depending on the concentration of sugar present in urine. Benedict's test. Materials required: urine sample in a test tube, Benedict's reagent, burner, test tube holder, and a dropper. Procedure: using a dropper, take a small quantity of Benedict's reagent. Now, add the reagent to the test tube containing urine sample. Boil the sample over a burner for 2 minutes, holding the test tube firmly with the test tube holder. Keep shaking the test tube as it is being heated. A brick red precipitate appears, indicating the presence of sugar in the urine. Conclusion. On boiling the urine sample with the Benedict's reagent, the cupric ion present in the Benedict's reagent is reduced by the reducing agent, sugar, to form a brick red colored precipitate of cuprous oxide. Therefore, sugar is present in the urine sample. Felling's test. Materials required: urine sample in a test tube holder, Felling's solution A, Felling's solution B, dropper, and a burner. Procedure. Using a dropper, take a small quantity of Felling solution A. Now, add the Felling solution A to the test tube containing urine sample. Using a dropper, take a small quantity of Felling solution B. Add Felling's solution B to the test tube containing urine sample. Boil the sample over a burner for 2 minutes, holding the test tube firmly with the test tube holder. Keep shaking the test tube while heating. A brick red precipitate appears, indicates the presence of sugar in the urine. Conclusion. The cupric ion present in the Felling solution is reduced on boiling by the reducing substance sugar to form the brick red colored precipitate of cuprous oxide. Therefore, sugar is present in the urine sample.
So is it clear to you? Benedict solution test.
Normal level.
What is the difference between the Benedict and
sugar through the urine? Sugar then starts to appear in urine.
Benedict's and Felling test?
Burner, test tube holder, and a
Benedict's,
dropper.
how Benedict is done and
Procedure.
how Felling
Now,
test is done.
add the reagent to the test tube containing urine sample.
So what are the steps?
Of Benedict's reagent. Now, add the reagent to the test tube. Using a dropper, take a small quantity of Benedict's reagent. Now, add the reagent to the test tube containing urine sample. Boil the sample over a burner for 2 minutes, holding the test tube firmly with the test tube holder. Keep shaking the test tube as it is being heated. A brick red precipitate appears.
So this is the difference. Benedict reagent is only added once to the urine sample, and Felling A and Felling B are added consequently one by one in the urine sample, and then both are heated. And on the basis of the color observed after the heating, we can tell what is the amount of the sugar which is present in the urine sample. Okay. Now simulator, we will see through simulator what is there, how we can do it. So Benedict test we are doing. Two sample, 2 ml urine sample is there, Benedict reagent is there, flame is there. First we will add the Benedict's reagent. Okay, we have added. So now we will put the this on the flame. Okay, we'll put this on the flame. Why it is not going on the flame? Okay, first we have to light the flame and then we have to put the test tube on the flame. For 2 minutes we have to flame it. It is turned green. Okay, it is turned green. So that means glucose is present in our urine sample. Okay, now we will do Felling test. So Felling test we are doing now. So पहले Felling solution A डालेंगे इसमें. So we'll put Felling solution A here in the urine sample. So first we put Pelling solution A, then we will put Pelling solution B, then we will flame it. We are flaming it, we are giving heat to it. Now we'll see what is the color change. So it is no color change is shown here. That means the absence of sugar in the urine. Benedict test is positive, so Felling test should also come positive. Then why it is not coming? It should turn into green. Yeah. Now it is turning green or yellow. We can say that if it is giving us yellow color or color is changed, that means the urine sample is containing the glucose. Okay. So I hope it is clear to you all. Now we'll do Viva voice. So let us see that whether you have understood the practicals or not. Try to give me the answer. What is the average production of urine in adult human? You can give the answer in chat or you can tell the answer orally also you can tell me. What is the average production of urine in adult human? 0. 0.5 liters, 1 liter, sorry, 10 liters, 2 liters or 5 liters. It's not minus 2, it is just dash. Dash 2 liters, dash 10 liters. So what we have studied in theory was 2 liters per day. Now what is the presence of glucose in urine called? इसमें से कौन सी टर्म बोलते हैं ग्लूकोज की प्रेजेंस को? यूरिन में ग्लूकोज की प्रेजेंस होती है तो ग्लूकोज यूरिया, रीनल ग्लूकोज, ब्लड ग्लूकोज, ग्लूकोज यूरिन, ग्लूकोज यूरिया. Which test is used to detect presence of sugars in urine sample? Benedict test, we have just studied. Which pigment gives light yellow color to the urine? Which pigment gives light yellow color? Albumin, chloroplast, melanin, urochrome. It's related to urine. Word is urochrome. What is nitrogenous organic substance present in urine? Nitrogenous organic substance कौन सा वाला है यूरिन में? यूरिया. उसका नाम यूरिन भी इसीलिए पड़ा है. Glucose start appearing in the urine if glucose level in the blood exceeds. किससे ज़्यादा होने पर ग्लूकोज आने लगता है? 160 to 180 मेगा मिलीग्राम पर डेसीलीटर. On boiling the urine sample with Pelling reagent it forms. Pelling reagent से रिएक्शन कराकर जब बॉईल करते हैं तो कलर्ड क्या बनता है? कलर्ड प्रेसिपिटेट ऑफ ग्लूकोज, कलर्ड प्रेसिपिटेट क्यूप्रस आयंस, कॉपर सल्फेट क्या बनता है? कॉपर सल्फाइड, क्यूप्रस ऑक्साइड. अब देखते हैं, हम सबमिट कर देते हैं आंसर्स को, देखते हैं कितने सही हुए हमारे. So first is correct, 2 liters average. What is presence? Glucose urea. Which test is used? Benedict test. Which pigment gives light yellow? Urochrome. What is nitrogenous organic? Urea. Glucose starts appearing 160 to 180. And on boiling the urine sample with Felling reagent, a colored precipitate cuprous oxide is there. So I hope this practical is clear to you. So your work of today is to do this practical in your file. We have done till now this was our third practical which we have done. Okay, so this was our third practical. So we I hope this is clear to you, and you will do it in your files. So I'm ending the class. You do this practical and show me by afterwards.